All right, welcome everyone. Good afternoon. It's a pleasure to have you, and I'd like to thank everyone for joining us today. I'm Dr. Tom Herzog. I'm here with Dr. Matt Powell, who's going to serve as co-moderator with me for this afternoon symposium. And we're looking at truth or perception, demystifying emergent data in the ever-growing competitive landscape for endometrial cancer. So it's great to be here. I hope all of you have been able to get beyond the convention center and see some of Dublin as well, my hometown. So hopefully you've been able to enjoy it a little bit. All right, so first of all, I want to thank the IGCS, GOG Foundation, and GOG Partners. And I'd also mostly want to thank GSK, who've made this possible with their support of this symposium. So we've all come together and to put this on, so thank you. So we are joined by an esteemed faculty, and I think we'll start at the far end here. We're going to have everyone introduce themselves, make sure the mics are working, make sure we know who you are and you know who you are. Otherwise, this is probably not going to go very well. Dr. Mansour. See? You're not doing that.
I don't know who I am, but my name is Mansour Mirza, and I'm a clinical oncologist from Copenhagen University Hospital. And thank you again for inviting me. It's such a pleasure. I would just say that 2 years ago, if you asked me something about molecular classification and what we have done, probably I won't be able to answer any of these things which I'm going to talk today. So we were in Stone Age just 2 years ago. We have changed everything.
Yeah, good afternoon, everybody. Rob Coleman, G1 oncologist in Austin, Texas. And I'm here next to Dr. Mirza to make sure that he remembers what he's supposed to tell you about from the Stone Ages.
Hi, everybody. I'm Isabelle Recocard, medical oncologist working in Lyon, France. Thank you so much to be there. So many people. We have a lot of things to do this session, and I'm happy to be part. I'm the woman.
All right. Yeah, good morning. And I'm Dr. Matthew Powell. I'm a gynecologic oncologist from Washington University in St. Louis. Excited to be here this morning, and we really do hope to make this interactive and get everybody involved. So there are microphones placed throughout this, and we'll be having questions throughout
I know
the talk.
it feels like morning, but it's really afternoon.
Oh yeah, good afternoon.
Yeah.
Good morning somewhere.
So I have a faculty that's disoriented to place, person, and time, but other than that, we're good to go. So let's rock and roll. These are disclosures. I'm not sure who wins. Oh, it looks like Dr. Mirza wins. Okay, good.
That's just page one.
That was the best. Dr. Oza yesterday, for
5
those of you
minutes.
who were in the session, he said, I've been given 5 minutes to do my talk. He goes, Dr. Mirza can't even get through his disclosures in 5 minutes. So I thought that was, I stole that from Amit. Oh, these are our learning objectives. I'm going to sort of summarize them. You can read them up on the screen here. But we're really going to be looking at some of the key data sets that have come out over the last year or so. It's been incredibly exciting over the last 18 months. And you know, obviously, the Ruby trials, 018, 10, DuoE, B21, and there's many other ones we're going to talk about. But we want to do so within the context of looking at some of the design complexities that are within these trials that really, I think, if you take a deeper dive, can impact how we interpret the data and how you go into clinical decision making. So I think how we're doing this, I think you'll find fruitful. I hope you will. We want to look at some of the new biomarkers that are out as well. So obviously we want to think about immune biomarkers, but we also want to think about, you know, indications for PARPs perhaps, some of the newer treatments that are available, and even looking at P53 as well as P53 wild type as a biomarker for some studies, right? So lots going on there. And then most of all, we want to engage with you. So we want to have this session be interactive. We want to allow time for question and answer. And we really encourage you to come up to the mics. And the group wants to be challenged. They've been saying all morning they want to be challenged. So let's not disappoint the group here. So they're really excited about this as we move forward. This is our agenda. As you can see, we're going to start off with looking at some of the emerging data with Dr. Mirza, and then we're going to talk about biomarkers with Dr. Ray Kakad. And then we're going to look at Dr. Coleman's ability to put all this in, or just confuse you with statistical terms. But he's going to try to make sense of some of the trials and look at some of the nuances and some of the things maybe we should be thinking about. And then we're going to bring everybody together and hopefully have a discussion with you and using the mics and questions and so forth. So, all right, let's kick it off. So our first speaker is Dr. Mansour from Copenhagen.
Thanks again. I think it's a great pleasure to be here and discuss. We are changing our guidelines every second month at the moment. The data, that amount of data we are seeing which is coming in, which is changing the way we are understanding this disease, is amazing. And we are privileged to be part of this. I like this slide to start with what we had, that is adjuvant radiation therapy for early stage. It clearly shows that there is a benefit of local control, not really in this meta-analysis showing benefit in the overall survival. Today I would call it a sad slide, pathetic slide. It was the wow 2 years ago, is the best we could do with the chemotherapy, carboplatin paclitaxel, which has shown in the stage 3, stage 4, and first relapse disease that it is really good. But look at in 3 years how many patients are alive, which was the reality of this disease just 2 years ago. And we have changed that. The best thing what happened was a decade ago when we saw the TCGA data, and we found out that this is not just type 1 and type 2 tumors. This is a very heterogeneous disease. And I would question that now we have 4 subgroups, molecular subgroups, and I'm sure Isabel will allude to that. But 4 is, I would say, kindergarten. It's a much more heterogeneous disease when it comes to molecular classification. The best part we have seen is that if you look during different tumor types, the number of tumors, number of patients which will have benefit from immune checkpoint inhibitor is highest in endometrial cancer. So that's why everybody jumped onto this and started working on this side. We know that there are about 30% of the patients who are mismatch repair deficient, which are very, the immune therapy is very effective, but we also have the rest of the tumors, which are sort of a cold tumors, and there we have to make them more hot by addition of chemotherapy, addition of PARP inhibitors, and all these is coming up, and we have results of most of these trials today. Remember when we planned, that's one thing I want to say. Remember when we planned these trials, we had no idea where we are up to. If we had designed these trials today, we will design it much differently. We will be much more smarter to design. So most of these trials we designed, I would call it dinosaur trials, dinosaur design. But also, the thing is, our success had so quickly changed our standard of care that the trial which were started earlier, where standard of care is something else, it will be very difficult to interpret the data. Third thing, the trials, all these trials which I'm going to show in the first line, they look alike, but they are not alike. They are different. There are differences between them, and I would call them complementary. They are adding information to each other. So we have to look at them as a collection. It's not comparing against each other, but adding more information. Some has carcinosarcoma, some has stage 3, some has this and that. So remember, and when you start, which population you take in the adjuvant therapy. So all this is complementary, okay? Like it or not, the upper line is the TCGA classification. You have POLE, DMMR, NSMP, P53mu. Why my POLE is very small? When you look at the TCGA, it says 7%, something like that. But when we look at the stage 3, 4, and first relapse, these are the trials done, these patients who are POLE biologically do not come stage up to stage 3, 4. They are mostly in stage 1 and 2. So if you do this in stage 1 and 2 probably, and Isabel will definitely talk about that, in there. So that's why it's a very small population. But this is just the beginning. I would say it is DMMR and everything else. And what is everything else? That's a Jurassic Park. I call it non-DMMR. MMRP is no classification. That's not a classification. It's a non-DMMR, and there you have P53 wild type, P53 mute, you have HER2, you have PD-L1 expression, you have ER positivity and negativity, and so on and so on and so on. So this is just the TCGA classification is just the beginning, and we are getting, have targets which will be coming in, and we will have results coming in every year to change our standard of care. Just to start with everything, when we start in the late line, second line, and in second line we had established that single agent immune checkpoint inhibitors, here dostarlimab and pembrolizumab, both approved on both sides of the pond, have shown single agent very strong activity with a response rates of 45–50%. What is the response rate of single agent chemotherapy here? That's what standard is, 10–15%. So that's why we got approval. And the first phase 3 trial ever done with immune checkpoint inhibitors in endometrial cancer was Keynote 775. It was done in second line, where patients, all patients, regardless of their DMMR status, all patients were randomized to receive either chemotherapy, standard of care. In second line, it's single agent doxorubicin or single agent weekly paclitaxel against pembrolizumab. So added TKI to enhance the efficacy of immune checkpoint inhibitors in the so-called cold tumors, the pMMR, and all primary endpoints were positive. Responses, PFS, in the pMMR, in intention to treat. If you look at the OS in the pMMR, everything was positive. So this was the first proof that we're really moving in the right direction. We're moving the needle to the right direction. Why we have to wait for immune checkpoint inhibitors to second line to give the treatment, that they have a place up front. So that's why we started, I would call, tsunami of phase 3 trials in first line. And we're asking 3 questions in these trials. The first question we are asking is, our standard of care for stage 3, stage 4, and first relapse is carboplatin paclitaxel. What if we add immune checkpoint inhibitor on top of that? Is it better? So first 3 trials are asking in that stage. And then in the EN11 trial, we are going even earlier, the B21 trial, even earlier, in stage 1 and 2 disease, or stage 3, 4, no residual disease. If you have high-risk population, can we do better by addition of immune checkpoint inhibitors? And I'll come back to that. Second question we started asking, on top of immune checkpoint inhibitors, so these trials were planned in the beginning, so that's why I said the designs could have been different if I did it today. The second was, the 2 trials we did were on top of adding immune checkpoint inhibitor. If we add PARP inhibitor on top of that, is it going to be even better? If at this time I will do the trial, I will probably select the population where I do the trial. It is done in whole population, and I'll come back to it. These are the trials we are still adding to carboplatin paclitaxel. The last trials are asking a very innovative question: can we get rid of carboplatin paclitaxel in first line and wait till relapse, and then they can get that treatment and replace it? The first trial, the LEAP-001, which has the results, it is a copy of the Keynote 775 just up front. That means that here you have a very strong comparator, not single-agent chemotherapy, but carboplatin paclitaxel. And I'll come back to that. The 2 last trials, we haven't got results yet. I felt at the time we started, these were the most nicest trials we did, biomarker-driven, only in the DMMR population. Either give carboplatin paclitaxel, which is standard of care, or just immune checkpoint inhibitor. In one, pembrolizumab, other dostarlimab. The problem is that we have overtaken our success has changed our standard of care to such an extent that carboplatin paclitaxel is not standard of care in DMMR anymore, and I'll come back to that as well. So what I'm going to do now is I will pool all these trials results together and put it according to the molecular classification, what we have learned. today. So first, let's look at the easy part, which is a home run, which is the DMMR population. And the DMMR is, as I said, it's about 30% of the population. And here, first I will show the early stage disease. That is stage 1, stage 2 patients who are DMMR with high-risk classification, and stage 3 and stage 4, no residual disease. These patients were entered in a large trial of 1,100 patients, Endometrial 11, Keynote B21 GOG 3053. That trial tried to answer the question. Of course, we always separate, stratify according to the DMMR status. And when we look at the whole population, the trial is negative. And one can ask the question, why it's negative when all other trials we have seen are extremely positive? Probably several reasons. First of all, probably adjuvant therapy where there is no tumor left, how immune therapy is going to biologically work there. You really need to have something. And that's why if you look at the chart on the right, you see the DMMR population. In DMMR population, you have a risk reduction of 70%, hazard ratio of 0.31 in this, which is which is the same, I would say, magnitude as you see in the more advanced. Probably here because you have very sensitive tumors. If you have a few cells left, you have efficacy. So I think I would look at this trial. Overall, it's negative, but I really hope that our regulators will understand that the biology counts here. It's not the stage of the disease, that if there is a DMMR tumor, I will take the next one to that. These are the trials, 4 trials done in stage 3, stage 4, and first relapse patients, chemotherapy, and then added immune checkpoint inhibitor. Here you have 2 of the trials with PD-1s, 2 PD-L1s, all are extremely positive. Look at the results. Look at the Ruby trial. First 6 months, there are some progressions coming, about to 30%, and then there are no progressions. It's all flat. Nothing is happening there. We have 2 years cut off. Nothing is happening there. A risk reduction of 72%, which is unprecedented. You have increased the PFS from 15% to 61%, which is really amazing. And you see all other trials, the same results. If I remove the names, probably you won't recognize which graph is to what. What this is telling us, this is extremely sensitive disease, the DMMR population. And then now we have also the early stage 1, 2, and 3 from the Endometrial 11. Altogether, my take is, although we need the regulators to approve in the early stage, my take is it is biology, not the stage of the disease when it comes to the DMMR population and efficacy of immune checkpoint inhibitors. If you have a high-risk patient in early stage, which is stage 1, 2, or 3, 4, no residual disease, and it is DMMR, and you are planning to treat that patient with carboplatin paclitaxel, you must add immune checkpoint inhibitor on top of that. And there's no question about the more advanced stage. You can see the results here. We have never seen such Kaplan-Meier curves in gynae oncology before. So what about the overall survival? Again, we do not have mature data. We have mature data only from the Ruby trial on overall survival. And here you can see that the curve is again flat. There are some events occurring in the DMMR population. These are the patients who entered the study when they were median age of 68. One has to die of something. So if you put, if you put your normal population, healthy population, and their survival curves on top of that, probably they will be the same. So what I'm saying is probably we have cured many of these patients by addition of immune checkpoint inhibitors. So I think this is something we should be proud of. The next question was, we added, you saw the next 2 trials which we added PARP inhibitors. And if I did today, I would not take them in the DMMR population. There is no biology telling us that PARP is going to help there. It's difficult to extract the data from the Ruby yet because we are still have to have more mature data. But in DuoE, you can see here in the PFS, there are two arms. One is with Durva alone and one with Durva plus olaparib. Can you see which one is what? No. You can look at the hazard ratios, 0.41 and 0.42 equal. PARP is not adding anything in the DMMR population, only toxicity. So don't add PARP in the DMMR population, that's for sure. The question now rises is, if these tumors are so sensitive to immune checkpoint inhibitors, what is happening in the first 30% of the patients? Why they are progressing? This is the thing this is what we have to answer in the future. That's what we are working on. What are the reasons why they are progressing? Another question is, after 1 year, 15 months, nothing is happening. It's all flat. Probably they are cured. Why we have to give treatment for 2 years, 3 years, or until progression of disease? Probably we have to do the new trials to reduce the duration of treatment. There is no need to treat these patients for a longer period, but we need clinical trials. We cannot just change it before that. This was a very nice small paper, interesting, in the ESMO, asking the same question. So if I put I have taken the methylation and non-methylation away because Isabel will take it. I don't want to overlap. But if we take the what we have now, there is unprecedented benefit of adding of immune checkpoint inhibitors to carboplatin paclitaxel in the DMMR population, regardless of stage, and it's the biology which counts. Do not add PARP inhibitor to these patients. And I would make it clear that the data from the two trials, Ruby and the GY018, shows us there is no difference in efficacy. It has good efficacy in the methylated or non-methylated tumors. I think we will have more granulation from Isabel later. Let's move to the MMRP. That becomes much more complicated, extremely heterogeneous group. I'm not going to talk about poly-mute because we had only, let's say, in Ruby trial, we had 5 patients with poly. None of them has progression. In the Ruby Part 2, we had 5 patients. 1 had progression in the placebo arm. You cannot say anything else. I would say that I should not treat these patients because it's very difficult. It's stage 3, 4, and first. We have to have much more data to take that decision. But it looks like that these patients are doing very well. The most important is the POL-E wild type, where you perform the P53 analysis, and you have P53 wild type patients, which are called NSMP. Another Jurassic Park, you have ER positive, ER negative, you have a little bit of PD-L1 positive, and all that mix up. And we have to separate them in future from each other. And the P53 abnormal, again, a very heterogeneous disease. Here you have a lot of HER2 positivity, carcinosarcomas, other possibilities. So this is a very, very heterogeneous group. We have to divide them in future. And when we look at the overall survival, here, I won't say it's a big wow. It's showing that you are benefiting the patients. You can see at the progression-free survival, the hazard ratios are improving, 0.76, 0.54, and so on. So there is a clear, modest, but it's a huge group, to 75% of the patients. There is a modest improvement in the whole population. But this is such a heterogeneous population, we have to figure out who are these patients. When we come add PARP inhibitor, here you start seeing some difference. Look at the DuoE data that the PFS I have highlighted here. The hazard ratio of 0.77 with addition of durvalumab alone. When you add the olaparib, it becomes 0.57. So there is definitely something happening here. And we saw Shannon presenting the molecular classification, and Isabel again will come to that. I have to rush away from it. So the next question is, which of the MMRP patients may benefit from immune checkpoint inhibitors plus chemotherapy? Sorry, this slide should not be here. This is here. So if I say that patients who, right now the evidence says that we have benefit of addition of immune checkpoint inhibitors plus minus PARP inhibitors in the MMRP as well, not as huge as we see the other one. The POLE, I have not discussed. When it looks at the P53 abnormal, we see a clear benefit. When it shifts to NSMP, we see modest benefit in that. I had this slide added into it, and this is to tell that how difficult for us it would be to find out overall survival benefit. So these are the patients in the RUBY Part 1 who have all of these patients who ended up in the relapse receiving immune checkpoint inhibitor alone or with lenvatinib. So these are not whole population, only the population which is receiving the subsequent immune checkpoint in second or third line patients. And even though they received it in second, and even though that the number of patients in placebo had a huge number who received immune checkpoint inhibitors, if you look at their survival curves, you still see a clear benefit of treating these patients up front. So what I'm trying to say here is immune checkpoint inhibitors should be given up front. I have -4 seconds. So now we have, I will skip that slide, just few slides on can we replace conventional therapy, the 3 trials, and the first trial, LEAP-001, was negative. So answer is right now, no, I don't think so, and we don't have data on these 2 trials which are there. So I will take the key takeaways here that I think we have done amazing progress in endometrial cancer by addition of immune checkpoint inhibitors. In DMMR, it's a done deal. In the MMRP, we have to make more granulation. But until then, if you look in the NSMP or in non-NSMP, the P53 mute, you see high benefit or modest benefit. So it's still better than carboplatin paclitaxel. So until we have something better, we have to change our standard of care. But we have a longer list of questions after that, how we are going to move forward, and we are doing that. Thank you so much.
All right, it's my great pleasure to invite up Isabel now that she's going to really answer all those things that Dr. Mirza failed to answer during his talk. No, that he actually alluded to. There's going to be great clarity on the biomarkers. So, Isabel, thank you.
Thank you so much. Considering biomarker, I would say that it is probably our grail. Why? Because when we are in our routine practice, managing our patient, and when they are coming in our consultation, we want to know what is the prognostic of our patient and which kind of tool we can use to be sure that the treatment we will manage will be the best one for our patient. However, we have to say that sometimes it could be also some kind of deception, and we have to be careful on that. Now moving to, thank you so much. Moving to that, we have to make some difference between what we call a biomarker to predict the prognostic of the disease and the biomarker who predict the efficacy of the treatment. That is not exactly the same. And right now, if we are discussing about endometrial carcinoma, there is some strong biomarker, and DMMR and HER2 are probably those who are able to really predict the activity of the treatment, but there is some other who have to be exploratory. It's about homologous recombination deficiency, about P53 status, PD-L1, TMB, hormonal receptor expression. But there is also new biomarker, perhaps coming soon, looking to what's happened in terms of new treatment in endometrial carcinoma as TROP2, folate receptor, but also B7H4 and MDMD. So moving to the strong biomarker, the first one is really the mismatch repair deficiency. We have a clear definition and clear recommendation from the pathologist and ASCO to report this status in terms of immunochemistry and NGS. And you have to remember that TMB should not be used as a surrogate of DMMR. I will come back on that later. We have an algorithm, and it is very easy in our routine practice using immunochemistry And if it is not so clear, we have the possibility to move to PCR or NGS to be sure about the real status for our patients. The first question you can ask is, do we see some exact similarity between DMMR and MSI status? There is this publication, including more than 600 tumor samples, and you can see that there is a good overlap between the both testing. There is some discordance, and in general, the discordance is in line with what we call now the subclonal loss of MMR, and in general is in line with the methylation of the MLH1 promoter. But the correlation is good. The second question is there one who are able to better predict the activity of immune therapy comparing DMMR status or MSI-I? This publication from last year seems to reveal that NGS could be a little bit better to predict the activity of immune therapy compared to immunochemistry. However, you have seen the recent presentation from Shannon Restin for DUO-E. I'm not sure it's the reality. It's one publication. Concerning the fact that MSI-I or DMMR is able to predict the activity of immune therapy and could be reproducible, the answer is yes. How I can tell that? Look at the other ratio in the different clinical trials. It's always the same. So we are sure that this testing is able to predict the activity of immune therapy. The question is about this subgroup of patients in the different clinical trials where we are not able to make the demonstration they are progressing in the same rate than the patient who receive only chemotherapy. How we can explain that? Is a question of misdiagnosis, is a question of subclonal DMMR status, we can discuss on that later, or is there any particular molecular subgroup who can explain this difference? Looking to what we have now in terms of clinical data, it's not seen there is a clear difference between methylation and mutation. And coming from RUBY or coming from the ENERGY trial, you can see that there is an activity of immune therapy in any subgroup, and this activity is really strong. However, I will be a little bit more cautious for the future. Why? Because we have several publications, and this one is really interesting, where we see that the molecular landscape in the promoter hypermethylation group versus germline mutation versus somatic mutation, you see is not exactly the same. If you look, for example, to JAK1, it's much more involved in the hypermethylation group, where we know that it can be something able to develop some resistance to immune therapy. In the other hand, we see more HER2, more HER3 amplification in the germline subgroup. And if you look to the complete publication in terms of TMB, the TMB median level is not exactly the same in the different subgroups. And the immune landscape is not always completely overlapping in terms of CD8 positive population. So right now, there is no difference. I agree with Mansour. I will be interested in the future with a longer follow-up to confirm if the magnitude of the benefit is exactly the same. Now coming back to HER2. HER2 amplification or HER2 overexpression is a very interesting program in endometrial carcinoma. First, because the prevalence is not so low. You can see on the slide, particularly for the carcinosarcoma, but also for endometrial carcinoma in general, the prevalence is between 6% to 40% if we look to the 2+ population. The good news is that when we look at the molecular subgroup and the HER2 overexpression, you can see on this paper that clearly HER2 expression is observed in the P53 mutated population. And so in terms of current algorithm, it's easy to reduce the number of patients where we have to test HER2 in this P53 mutated population. And why we need to do that is because we have right now treatment who are particularly efficient in this population of patients with overexpression. We have the TDXD, but we have now several clinical trials coming soon who we will test this kind of drug for these patients. If there is some limitation, Yes, of course, as usual. We have seen that some difference in terms of agreement between the local testing and the central testing in the publication. There is also some difference when we are using the breast algorithm compared to the gastric algorithm. But when you look at the detail in the gynae cohort, you see that finally the difference is for the 2+ population, where some of them with the central review is coming to be 1+. Do we have the possibility to improve our capability to identify this patient? Of course, blood sample is always very interesting for our patient instead of tumor tissue. And we have seen very nice results using the ctDNA to predict the activity of EDC targeting HER2. However, you can see on the slide that we will probably reduce the number of candidates. So right now, it's very specific, unfortunately a little bit not enough sensitive to identify all our candidates. So if we make some general conclusion for HER2, there is overexpression and amplification correlated to p53 molecular subgroup, particularly in the serous and the carcinosarcoma subgroup. There is some discordance in terms of rate, particularly in the HER2 2+ and 1+. However, we continue to learn with the clinical trial ongoing. The ctDNA testing may be help to identify patient, but not yet to be a substitute. Now moving to p53. p53 is part of our molecular classification, is a prognostic biomarker. We see some activity to predict the activity of PARP inhibitor, of radiotherapy also, and there is some correlation between p53 and HRD. Considering p53 mutation in endometrial carcinoma, there is a good concordance between immunochemistry and NGS. So it is something quite published several times, and we see some good activity that immunochemistry could be enough for this patient before to move to NGS. Is p53 status able to predict the IO activity? When we see the first result presented by Mansour for the RUBY trial, we see a huge difference in the p53 mutated population using dostarlimab plus chemo versus chemo alone. However, 2 days ago, what Shannon reports in the DUO-E trial looking to the activity of durva in combination with chemotherapy in the p53 mutated population versus wild type is not so easy, and we are not sure that in this trial we see exactly the same results than we have seen in the RUBY trial before. Is this p53 status is able to predict the PARP activity? There is again several subgroup analyses. We see that in the RUBY2 part a clear benefit with another ratio of 0.29 for the niraparib used in this cohort of patients. Florence Joly with UTOLA also report some activity of olaparib in the p53 mutated population. It's just a randomized phase 2. But again, we see some recent data coming from the DUO-E publication, where we see that the other ratio is not exactly the same, 0.47 for the p53 mutated population, but 0.71 for the wild-type p53 population. That means that it is not already clear for me. Moving to the HRR mutation, there is some data in the literature considering that the number of patients with homologous recombination repair deficiency with mutation in the list of the gene is a little bit more than in ovarian cancer. However, we have to be careful. The list of the gene is changing over time, and I'm not sure that ERID1 could be considered as able to predict the activity of PARP inhibitor. Is this a group of patients have some capability to predict the activity of PARP inhibitor in endometrial carcinoma? It's not so clear. Looking to RUBY and also looking to the DUO-E trial, we don't see a strong capability to predict the activity of PARP inhibitor. HRD could be something interesting. There is several publications. This one is using RNA-seq signature to predict the HRD. It seems to be prognostic for endometrial carcinoma. There is also some functional HRD test used in endometrial carcinoma population, and you can see that This group of patients in general is serous endometrial carcinoma, p53 mutated, or with some BRCA and BRIP1 mutation. However, do we know if HRD test is able to predict the activity of PARP inhibitor? It seems to be the case for the UTOLA trial exploring olaparib alone. However, it is a post-hoc analysis and it is a small phase 2 trial. What about PD-L1? PD-L1 expression is already reported several times in endometrial carcinoma, and it's a huge number of patients, 77% in this publication. What about the role of PD-L1 to predict immunotherapy? We have seen in the Garnet post-hoc analysis that finally the PD-L1 expression is not really enough strong to predict the activity of immune therapy in this population, particularly you imagine in the DMMR population. Is there any data coming from the large phase 3? The NRG report that there is no clear indication to look at the PD-L1 expression to predict the activity of immune therapy in the DMMR or in the PMMR population. We see exactly the same in the ANTEN trial, looking to atezolizumab in the first-line setting. Only the DUO-E publication at the first publication presents, oh, I'm sorry, I lose my slide. I can continue, but I'm not sure it will be easy for you. Okay.
Just give it a second. It'll come up.
Okay. I'm sorry. Where I am?
Oops. That's a long way.
Mansour, you got to start over.
Before?
He's doing it. He's
Okay.
doing it. Don't do anything. We're good.
The message for PD-L1 before we receive the slide is that also in the DUO-E we see some data considering that the PFS will be.
What you can do is you
Okay,
can pull it
this
here
one.
and click.
Okay.
There you go.
It's fine? Okay. We see some benefit to in the PD-L1 positive population compared to the PD-L1 negative. You have to see that the number of patients in the PD-L1 negative is very small. And when you look at the other ratio in the most recent publication from Shannon, it not seems to be easy. And you have to remember that the expression of PD-L1 need a lot of expertise in terms of pathologists, and looking to the tumor sample we are using, I'm not sure that it is a strong biomarker for the future. Do we anticipate that TMB could be something interesting in the future? You remember this publication in the Lancet 2020, where we have seen that TMB is something important in endometrial carcinoma, and it's not able to completely overlap with a DMMR population. And again, coming from Garnet, we have seen some interesting data. If you look carefully, there is more activity in the TMB high than in the TMB low population. Also, we see that in the TMB high, we see the same response rate for the patient with a DMMR status compared to the patient with a PMMR population. And in the DMMR population, if the patient have a TMB low, the response rate is a little bit less than for the other. This is interesting for the future, I think. Coming back now to the NSMP population. By definition, there is no biomarker. Not sure. We have the ER/PR positive expression that it could be something interesting for hormonal treatment, and I hope in the future for CDK4/6 inhibitor. But we have also other biomarker, and I hope that in the future it could be interesting. Now we have to remember that there is new drug coming soon. We speak about EDC, but there is also selinexor, for example. There is some other interesting drug, and I hope that there is new biomarker. And finally, we need to remember that this is a very heterogeneous population where there is not only p53 mutation, not only DMMR population or poly mutation. There is also actionable targeted alteration as KRAS, as P3K alteration, as FGFR2. And we have now drug in developing on this particular setting, and I hope that this new target will be biomarker for our patient. So just to finalize, we have validated biomarker able to predict the prognostic, but also able to predict the activity of the drug as DMMR, as R2. There is exploratory biomarker, we need to work on that. I would like just to emphasize the fact that DMMR, MSI, endometrial cancer continue to be an heterogeneous population regarding the mechanism of mismatch repair, and there is potentially secondary alteration, microenvironment, that we need to continue to work on that. For me, it's not clear how P53 abnormal tumor may respond or not to immune therapy and perhaps PARP inhibitor. We need to continue to work if we want to conclude on the HRD testing, and HER2 should be integrated in our practice if we want to help our patients. Thank you so much for your attention. I'm late, no?
Yeah. All right.
I'm late.
Isabel, thank you. That was wonderful. It was a great look at the biomarker impact on the endometrial cancer space. And so our next speaker is Dr. Rob Coleman, who's going to unravel endometrial cancer data complexities and shed some light on our guidelines and how we got there.
Okay, thank you. Thank you. Appreciate the invitation to speak. So we thought we would kind of close this session before we get to the Q&A with a little different kind of bend on the topic. And I realize that many people in this room are not from the United States, but I thought it would be insightful to take a review about how the regulatory process works to put these studies in context. Because a lot of questions that come up, and I loved Isabel's talk because almost every slide was ended with a question. Almost every slide ended with a question, right? Which then, what does it do? It makes you say, okay, I'm going to make a hypothesis, and I'm going to test that hypothesis. That whole process drives a lot of how we design the clinical trial. So I'm going to start off with a little bit of history and understanding of how the regulatory process works in the United States, because that does drive a lot of the data that you saw in these early trials and these late trials with the intent of bringing regulatory approval. Talk a little bit about some of the outcomes and how these hypotheses were addressed, and then how these end up in guidelines. Because that's another question that comes up. It's obvious if a phase 3 trial that's well designed and analyzed comes up with a statistically significant result, that it would end up in guideline. Certainly, maybe end up even with the regulatory approval, and in some places actually end up with paid regulatory approval. But what about all these other questions that were raised? How do subgroups and how do they end up in these guidelines that we often quote? So let me start off with a series of programs that the US FDA launched many years ago to provide an incentive to bring new compounds to the clinic. These 4 programs were launched over a period of about 20 years, and you can see the 2 first programs were this accelerated approval program and this priority review. And the intent there was to provide developers to bring early results of clinical trials into before the agency to evaluate their efficacy. And they challenged us to come up with surrogate endpoints that would be ultimately predictive of a long-term outcome. And they also helped to accelerate the timeline to get there. So this is what the priority review is, and there are other kind of components here with respect to fast track and breakthrough designation, which I won't talk about, but they essentially iterated on this whole concept of trying to bring new compounds quicker to the environment. And ultimately, this program ended up into formal guidance, and you can read this online. It has a lot of tenets that have continued to iterate over all these years now, about 30 years in the making. So the process that and I call this the FDA playbook because this is kind of what most of these trials that you just heard about are based on. There's this idea of a proof of concept, and that usually comes from trials that many of you do or participate in, to say, hey, there's a signal here. And then what we do is then say, okay, if there's a signal here, maybe we need to figure out exactly what's the dose and schedule and duration of therapy that we should use to bring that forward into further development. That's called dose optimization. And that part of this regulatory process has continued to also iterate, becoming much more complex and also much more informative. Ultimately, once we find the right dose, that gets expanded to have enough clinical data to get an early signal. And in many cases, that signal is based on objective response. In some cases where we see growth inhibition, if the asset prevents growth of tumor, we might look at a PFS endpoint. And frequently, these are single-arm trials, or sometimes can actually be even randomized trials. But again, they're done with limited cohort sample size to provide some inference on whether or not this is going to be a long-term win. Okay, now with respect to the way the US FDA is conducted, is that while that signal looks strong, the new rule is that a confirmatory trial needs to be actually enrolling and ultimately substantially enrolled when the new asset is brought before the agency to be considered for this accelerated approval program. So you'll see this, you'll feel this when we see in phase 2 trials, for instance, which have been launched and seem to have a strong signal, and you say, oh, hey, there's a phase 3 trial that's ongoing. That's the point. The point is that they want to evaluate this new information against a relevant randomized trial that would try to confirm that efficacy and show that it's better than existing standards. And then ultimately, that would provide then an opportunity to filing for regular approval. The other way to do this is to just launch a regulatory strategy right from the beginning. So you do a registration trial, formal phase 3, we got a dose and we just go for it. So we're not really interested in that early signal. And sometimes in a situation where a drug is basically growth inhibitory, there's no real strong evidence to get an accelerated approval, so they just go right into a formal phase 3 trial, and that's what that is. If there is an opportunity to see a signal, they may embed it into the trial itself while it's ongoing. And this has actually been, it's been emphasized, encouraged, I think, from the FDA, because it basically saves time and patient experience to address two questions at once, accelerated approval and regular approval. What we found in real time, what's happening in the real world, is that these trials, if they have exciting signals, is that they enroll very fast, and so we really don't save ultimately very much time. But this is another encouraged design. So these are designs that you will see frequently. You'll come to the meeting, you'll see a phase 2, then you'll see another paper like we saw with Ruby and DUO-E and GY018, where we've confirmed these trials in the larger sample that ultimately led to approval. So the FDA accelerated approval process was launched initially to promote the development of HIV drugs. So that was the initial, and the first drug that was this zalcitabine, which was brought forward and ultimately proved not to be effective. So it was granted accelerated approval, and the confirmatory trial was negative. But over time, because the tenets of accelerated approval are predicated on that the disease is serious and has to provide benefit over existing therapy, it was a natural fit for the clinical development of oncology drugs. And rapidly, that proportion of oncology to non-oncology drugs shifted in favor of oncology drug. We're now about over 80% of the new applications that are presented to the agency are in the oncology space. That's why there's so much activity. So there's several little nuances in this, and I go to the FDA frequently to talk to them about what are the benchmarks we need to achieve, how do we design our trials, and it continues to evolve. So because of that, it's interesting because we learn new things about the process. The problem is that we can't use precedent. So you can say, well, you did that in the past, and they're like, you're right, we did. But that doesn't apply now, So it's been a little bit frustrating to try to get a handle on these processes. But there are some very consistent tenets that do come up. One is this heterogeneous population. So while we may see evidence from a global trial that is enrolled in patients from all over the world, what the agency is interested in patients that are essentially what we call US-like, but have essentially the exposures and the opportunities for therapy pre and post-treatment that would be similar in the United States. So even though a global trial can help us with an accelerated approval, they're looking for a proportion of patients that are actually, quote, US-like. And this has to be accompanied with a safety database, and as mentioned, the confirmatory trial needs to be significantly enrolled. So has there been any benefit to this program? This is a paper that was just recently published, and I loved it because it basically says that this process that they initiated over 30 years ago has saved lives. So this is across multiple different tumor types on the right-hand side of this slide. These are the actual cumulative life years gained by the accelerated approval process. So how does this save lives? Well, obviously, many of the accelerated approvals may not actually go forward and improve benefit in their confirmatory trials. We've seen that many times, but many of them do. And so what that allows is an earlier exposure of a patient population that would have otherwise not seen activity in this of this new agent, and they would have gained. And so on the left-hand side of the slide, what you see in those blue areas are essentially the hazard ratios of having an earlier access to a drug before it gets regular approval. And those benefits have accumulated in life year gained. So I think this has been validation that the program not only helped to incentivize drug development, but that the drug development, especially for people who participate in it, provides benefit. So again, if you needed any encouragement to contribute to clinical trials, this is one of the strongest arguments. You could also say that because accelerated approval allows the compound to be on the market earlier, there's also a financial or a marketing potential benefit because that drug now can be prescribed as a standard of care pending the regular approval. Now, because of this timeline, in the past it had been quite long. The FDA has started to shrink it down so that the confirmatory trial needs to be substantially enrolled so that the timelines between accelerated and regular approval are essentially shortened, but it still provides that benefit and opportunity to patients. So the accelerated approval process is one mechanism that has been really good. What has come back to us more in a more difficult way is dose optimization. As I mentioned, I said, once you find the signal, you optimize the dose and you go forward. Well, that middle step is what this guidance document is all about. So this is the last, it continues. Again, these are, if you're not familiar with the agency, the US agency, is that they will publish draft guidance and leave it open for public comment. So if you have an opinion, they'll get it. I don't know if they'll read it, but you can actually apply it online, and you can put your thoughts into this. I do think they try to understand input from the general public and industry as to how these things might help provide stronger guidance for and actually optimize the system so that we get it right. And I'll just give you one idea one issue that's coming up right now frequently with the antibody-drug conjugates is like how long if you're going to give an antibody-drug conjugate in the maintenance setting, let's say for an endometrial cancer trial, how long is long enough? And if it's related, if the efficacy of the biomarker if efficacy is related to the biomarker, do you need the same dose for a highly expressing tumor as opposed to a less expressing tumor? And do you need to give it for the same duration? So these are the kinds of things that we think about and go into the design of these trials when we talk about duration of therapy. As Dr. Mirza mentioned to you, the 3 or now 5 trials that have been done in the advanced or first metastatic trials that he presented have different durations of the immune therapy duration. So is it 2 years? Is it 3 years? Is it progression? All those were addressed. They're never optimized. So the FDA is looking at this because it is a definite feature that could impact the tolerance or the safety of the compound. So this is just a typical strategy. As many people who know me know that I'm not a fan of the 3+3 design, but just to give you some idea that there's a dose escalation process that happens here, where there's an expansion usually to confirm that the dose can be repeated and it's safe at that point in time. And then what is now our new process for dose optimization is to do a randomized trial at the MTD, or what the dose is that you think is going to go forward, and the dose level below. And so there's some strategy to figure out what that dose level below should be. But we try to find out by doing extensive pharmacokinetic analysis that lower dose or the higher dose that you think is supposed to go forward actually provides the best response and the lowest and the acceptable toxicity. So once that's done, then we would then go on to that, as I mentioned, the regulatory strategy with a confirmatory trial. So I think Dr. Mirza mentioned that he shared with you these efficacy results, and he said, against standard chemotherapy, this looks good. Well, that's kind of the historical benchmark that in this process of a regulatory strategy to bring a new drug forward that we try to benchmark against. And so I have just some kind of reference points. But so if you do a single-arm trial and you see a 35% response rate, is that good? Well, all we can go on is the characteristics of the patient population and what we have for historical control. So in the past, what we've done is we've used these kind of like we developed these trials in the recurrent setting to be essentially against a well-trod standard, which would be single-agent chemotherapy. In endometrial cancer, this would be like a paclitaxel or an adriamycin-based treatment. And you can see that our expectations for response rates are around 10 to 15%. So those are the kind of the goalposts that we're trying to look for in our exploratory analysis to see whether or not there's a strong enough signal. And there's many, many examples of this that you just saw. So just in looking at the immune therapy, as was presented in the first lecture, you can see these single-arm trials that have looked at the efficacy by biomarker in these populations of patients with recurrent or endometrial cancer. And you can see that these all provided a strong signal in the dMMR population, but even a stronger signal in that pMMR population with len-pem. So these were all open-label, single-arm, forward-going trials. So with respect to how these got to where they are within the regulatory approval, you can see that in 2017 and 2019, we saw some accelerated approvals hit with pembrolizumab alone or in combination, and dostarlimab in 2021, and this most recently, TDXD. Now this provided us a kind of another question mark, because this is a study that was based on a pan-tumor indication with a relatively small cohort of patients, 40 patients. And what happened is that when this was evaluated across all tumor types, they felt that the efficacy was strong enough to provide this pan-tumor indication in the 3+, IHC 3+ for HER2. And this is the document that was published in last April. So that became an option for treatment. But if you look at the regulatory, the NCCN guidelines for this, there was no form, everything I just told you about having to have a substantially enrolled confirmatory trial has not been done. It has already entered into our guidelines. And you can see that avelumab and nivolumab, as two other immune checkpoint inhibitors, have also entered into our domain without having really any formal confirmatory trial in that process. And you can see also that HER2 2+ was allowed into this guidelines. So how did it get there? Well, this is the regulatory process and the guideline process are not necessarily completely connected. And so this was felt that patients should have access to it because in the 2+, we did see some efficacy that seemed to be better than available therapy as chemotherapy. Again, needing to be confirmed with follow-up trials, and that's what we're stuck with. So Ruby, GY018, DUO-E, Keynote 775 were all confirmatory trials that were predicated on confirming the efficacy of single-agent immune therapy in dMMR, ultimately in an pMMR population for Keynote 775, and as you know, those were all confirmatory. So why is this process so hard? Why is it so difficult? Well, one of the things is that we have very poor predictors of what's going to be successful when we get to the phase 3 setting. So this is some data that was published a couple years ago that looked at hazard ratio that were carried forward when they were formally evaluated in a phase 3 setting. So what you have on the x-axis are the hazard ratios for progression-free and overall survival in a phase 2 study, and then on the y-axis you have the hazard ratios for progression-free and overall survival in the phase 3 setting. And the lines represent unity. And so what you can see is that less than a third of the patients that show, a third of the studies that show a benefit in the phase 2 setting for PFS are actually confirmed, and only 2% for overall survival. So again, this gets to the point of looking at a subgroup, making a decision on the subgroup, and then hoping or inferring that it's going to be positive in the phase 3 setting. There are many factors that contribute to this disconnect between the phase 2 and phase 3 setting, one of which is what we share with you very frequently, which is post-progression opportunities. And as patients who have a longer post-progression survival after their first therapy, they have a lot of opportunities to get other treatments, including other treatments that may lead to a benefit in overall survival. So there could be a disconnect from the index randomization to the ultimate survival of overall survival. But I love this slide. I took this from Dr. Mirza, and thank you for sending it to me again, because I want to highlight that in the Ruby study, there was this issue of crossover. And certainly you can see there is, but I want you to just pay attention to the first subsequent and the second subsequent therapy. The noise between the first subsequent therapy and the second subsequent therapy, you can see how almost random it is, and how this could potentially obscure something that happened before the first subsequent therapy on the randomization. And so what we're hoping to do with this data going forward is to also put some timelines with this to help you show, to help kind of better illustrate how difficult it is to control for things such as crossover. Ultimately, you know, Dr. Isabel Ray-Coquard's presentation, or excellent presentation about biomarkers really hit home, because when we think about success of a predictive biomarker in the idea of a PFS to OS relationship, the trials that have won have all had predictive biomarkers. The ones that have not won are the ones that didn't. And so this, as you said, all work is not done, Isabel, when you finished your last slide. I think it's exactly right. We're not done yet. And again, this treatment effect can be quite impressive. Obviously, Mansour mentioned that the DMR status is one point to hit here. But what I wanted to get to is that there's ideal difference between the analytical endpoint and an inferred endpoint from what we saw in a subgroup analysis. And you saw Dr. Ray-Coquard mentioned about the issue of what are the biomarkers that are driving success for PARP, and these are the two trials. I'm not going to share with these again, but I want to get to one point here. And that's this issue about P53. So this, again, is a subgroup that has a relatively strong signal for, of an impact in the RUBY trial, but less so in the DUO-E trial. And I again want to give credit to this iterative process where a trial was done, the Endo trial, which suggested that there was this benefit in this P53 MMRP subgroup, even though the overall general results of the trial were less impressive. And so what they did is they said, okay, we see that this is the case, let's test it formally. And so this Export-EC trial is ongoing now to test the role of P53. So I hope you got a chance to see how the FDA programs have been structured and why that's important for drug development. I think this regulatory guidance has continued to help us iterate and make the trial designs much stronger. We definitely have a number of analytical strategies to help with our processing here, but we can't control for all of the issues that come up. And so these hypothesis-generating exercises should be that, hypothesis-generating, formally tested going forward. And I appreciate the attention, your attention. Thank you.
All right, fantastic. We have now about 20 minutes, and we're going to start to see if there's any questions from the audience. And as people are walking to the microphone, I'll follow up with Dr. Coleman. This inferred versus analytic. Can you expand on that a little bit? You went through that slide. I know you were seeing the red on the slide up here, or the timer. So,
I know.
But expand on that a little bit.
Yeah. So, so analytical basically means that when you design the trial to begin with, you've actually formed a hypothesis and then allocated enough alpha or error assessment to the hypothesis. So it's a formal question, and it's properly powered so that you can assess whether or not you can reject or not reject the null hypothesis. The inferred endpoint is essentially what we saw in those subgroups, and you'll see that there'll be frequently a hazard ratio for each one of the subgroups. And sometimes you'll see a p-value that's on the right-hand side of that. That p-value is not looking at the value, it should not be looking at what the p-value is for that subgroup. What it should be telling you is whether the subgroup has a result that's different than what was seen in the overall population. And if it is significant, then that means that there is a signal there, but still needs to be tested. So the inference is that there may be something there, but the formal testing is actually going to provide the analytical endpoint. That make sense? Okay.
Well, I want to expand on what Dr. Mirza said. Dinosaur trial you mentioned initially.
He's talking about him.
Is part of this, no, not him being a dinosaur.
Oh, yeah. Oh, okay.
Dinosaur trial. And part of that, when you look at the differences in how we design 018 and Ruby, you want to expand on what you meant by dinosaur? We didn't know exactly what we were doing at that point, and maybe some of the differences in trial design.
So yes, I think it's not only the Ruby and Duo. I would add also Duo-E and Ruby Part 2 in that. And for that reason, also the LiPo-1, all these trials, when we designed it, we had only one information that patients who are DMMR, they will have the highest efficacy. So we stratified according to that. But then we called it PMMR, which is just not PMMR. It's a non-DMMR, which is 10 different molecular subgroups with a different way they are going to react. Some will have efficacy of chemotherapy plus immune checkpoints. Other would have a benefit of PARP inhibitors, and so on and so on and so on. And why would I dilute, let's say, in the DUO-E or in Ruby Part 2 today, why would I dilute and treat patients with DMMR where biology doesn't tell us that it will work, a PARP inhibitor on top of that? The same is the case for estrogen receptor positive grid 1 and 2. What is the biology? So we now have to do much more cleverer trial. I'm not saying that we did wrong. We had no other evidence. We had to do these trials to figure out where it fits. If we never did it, we will be sitting here saying, oh, what about that subgroup and that subgroup? So I call them dinosaur trials, but they were necessary. We had to do them. These were the initial trials, and now we can move further and do much more intelligent trials, which are driven by biology, driven by biomarkers.
All right, well, please come to the microphone if you have questions, but I'll ask Isabel, what is your wish list when you look at all these trials that are just presented? What do you wish you had
her wish list is
from a biomarker standpoint on all these studies? What do you wish we had? I mean, I don't see any HER2 on DUO-E. I don't see, what do you think we should use to help parcel out all this? What would be your wish list to see published?
It is Christmas or it's not Christmas?
It's Christmas.
The first point, I want to go back to the dinosaur trial, and I would like to insist on that. Endometrial carcinoma is always observed in patients. The majority are elderly patients with comorbidity. And right now, we continue to have chemotherapy add to immune therapy also in the DMMR population that we don't have in colon cancer. And this is something that we need to take this responsibility. And now we need to work on that. We have this strong biomarker, and I'm sure that altogether we will be able to do something where we can try to avoid chemotherapy for this patient. This is my dream. Now, coming back to what you say, we have now the good opportunity to have several drugs efficient for our patients. We have immune therapy, we have PARP, we have EDC, we have new drug. We will not be able, and I don't want to imagine that we will give all the drug together for the same patient. So that means that we don't have choice. We need to continue to work on this biomarker, and I know that Rob explained us very well. We can't be immediately able to do the right biomarker to design the right trial, but working all together, doing some post-hoc analysis, then moving to the prospective way to validate, we will be able to decipher the population and to give the same opportunity to endometrial carcinoma patients as we see for lung cancer.
I think there is a balance. I completely agree. Balance between how narrow we would go or how broad we will go. An example is HER2 trials with ADCs. Are we going to take 3+ and 2+, or are we also going to take 1+? How we are going to interpret the data? I would like to have benefit for most of the patients, but that may be diluted by 1+. So this is the balance we have to find every time we design a trial.
All right. Still don't see any questions. Don't be shy. They won't necessarily have the answer, but they'll have the correct answer, but people will have an answer.
Yeah.
Oh, here we go.
Doesn't have to be right.
Hi, Mauricio Cuello from Chile. I have a question related with biomarkers and dipping back, because you're telling we have more information to better design clinical trials. But I'm thinking that you have a lot of information from those patients already part of the clinical trials. I meant biological information from the tissue you collected.
Right.
Exactly.
Why you don't look at that before
we are
moving
doing that.
forward?
Yeah.
That's what we are doing right now. We are looking, so The problem is there is a limited number tissue available, so we have to. That's what I think Matt asked. What is your wish list to Isabel? What is the wish list? What we want to see first, second, because at the end you have very, very little tissue left. So this is the way we are moving forward. Understanding scientific, Isabel, I think you can explain it much better, because I think this is the biggest thing we are doing right now in all these big trials. We have tissue available and we are working on the translation. Let Isabel explain if you would like to add.
No, the point is that there is this great opportunity. We have large phase 3, and I hope that we will be able to bring some a collaboration between the different groups, because with the number of patients included in the clinical trial, we designed the trial for the ITT population or for some subgroup. Now moving in the subgroup, as Rob mentioned, we know that the number of patients will not be enough. So one of the first things is to try to have some collaboration between the different groups to be able to have more tumor samples. Then the second point is, what is the priority? Is DNA, RNA-seq, immune chemistry expression. We can't be able to do all the things. And looking to what we say in terms of unmet medical need, probably the DMMR population, looking to methylation, for example, versus mutation, is very interesting, but it is the unmet medical need today, not sure. The poor prognostic population need always to be the most important.
I think there we have HER2 1+. I don't know if these are the patients who are progressing in first year, so on and so on. There are many more interesting things to look at.
I think the one thing, we've done biomarkers for years and years. I'm looking at people who've done spent a lot of their career doing biomarker work. We have a lot of prognostic biomarkers. Those are helpful in helping us design the trials for stratification variables. That's it. Most of those trials that have done that and have brought those forward as a predictive biomarker have failed. So I think on my wish list is to be able to hone down this hundreds of biomarkers that are obviously influenced. They're not just one factor. They're all related in certain ways. And I'm hoping now with better expansion of iterative AI technology and our ability to look at cell-free DNA, things that we can do without having to repeat biopsy everybody, that we would come up with more predictive biomarkers that can then be integrated into these trials in a much more efficient way. John.
Hi. John. Thank you for the great presentations. When we divide up all these uterine cancers into these distinct cell types, I almost feel like we're dealing with subgroups of rare cancers now. Man, how are we going to get the better design of these trials when we're almost feeling like we're in a rare tumor committee at the NRG now? Number two is, we're also making a lot of assumptions based on these exploratory post-hoc analysis, whether they're from PORTEC-3. I mean, I don't know how well balanced these groups are, and we're showing these curves here, and we're almost making outright recommendations without prospective trials, without validation. We don't even know if these two groups are even propensity matched. Right? It's kind of dangerous.
I thought about that in the master session we had right before that. The sub-analysis in PORTEC-3 showing chemotherapy for P53 mutated cancers look to be gospel.
Right.
It needs a confirmatory trial. I know there's a confirmatory trial that'll show us a little bit more, but it's not going to be perfect.
Yeah, you could have given chemo to all your stage 1 patients, and then the stage 3s, you didn't give chemo, so it was just a stage difference.
So that's the whole point, John.
It's dangerous.
It is dangerous because most of the time, as I said, if it's not a predictive biomarker, it actually has not panned out in formal analysis. And sometimes, like we've seen with some IO therapies in ovarian cancer, it's detrimental.
Yeah, well, thanks for agreeing with me. I need some answers.
So here's
Great questions, John. I told you they'll have responses. They may not.
No. Let me address it.
Let me address it. Sit down.
Let me address it because I do think that there's an opportunity here. So we look at those forest plots, and we pick out the one that we think has the most signal. We forget that every single patient who's enrolled in the trial occupies every single category. So if we're going to do these subgroup analyses,
Absolutely.
we need to control for the confounding other variables and try to isolate the single difference between those. It's going to be small groups, but at least you have a better handle on what's actually driving that signal.
And adding to that, I would say that we would be able to hypothesize on these subgroups, not to change our treatment, not to decide our treatment in standard of care. Maybe NCCN is going to do that, but that's not, happily I'm in Europe. But it's more to design the new trials, how we smartly design the new trials. And you are talking about rare disease.
Yeah.
Yes, you know, we are so fortunate that NGOT, GOG, EPGOT, we work so close together that we have whole globe, and we do these small subgroups of patients in no time. We will get the answers. So this is the beauty of working together.
But the other thing I was thinking is the answer tumor agnostic approvals? You know, just like in HER2, right?
Yeah, but so that has
Put all
accelerated
these
approval.
cancers
It's
together.
still going to be.
That's not approval.
It's not. It doesn't have regular approval, but that's fine because that looks to be a predictive biomarker for that agent. Yep, agreed.
All right. Tom, do you want to take us through this case?
Sure. It's a great question, John. You know, one thing about it, though, is that we're getting smarter all the time with these trials because these inferred, underpowered subgroups are setting up hypotheses that we can then test in the subsequent trials. And to your point, Mansoor, the ability to work together has made us much more efficient. For those of us older in the room, remember everything that had to be done on one side of the pond had to be recapitulated on the other side of the pond, sometimes at the expense of thousands of patients, our most valuable resource. So I think we are making some improvements there. All right, any other questions? Come up to the mic, otherwise I have to do a case. Okay, we'll do a case. A 54-year-old with stage 3C, grade 3 endometrial cancer. She was found to have, she was PMMR, no additional biomarkers were identified. HER2 was IHC 0. She had no medical comorbidities. She was counseled to go on a trial, but had refused. A surgery followed by CT scans show measurable disease in the periaortic lymph nodes, and she had a positive periaortic lymph node at the time of surgery. And you can see there at the level of the renals vessels. And she also has some indeterminate pulmonary nodules on CT after surgery. So anybody from the audience want to take a stab at this, or we can call on the panel here? So she'd certainly be eligible for some of the old trials that we've been talking about, right? So Ruby, for example. What would you do with her, Mansoor?
So this is a patient. Well, first of all, I need more information here. You are lacking information of the molecular classification.
It's Matt's case, so he'll tell you anything you want to know for a price.
What do you need to know?
You have $2 to spend.
You want
$2, I have $2 to spend.
Yeah.
Then I will use surgeon to ask if he will go ahead and see if patient is resectable. I'm not sure. There's an intermediate pulmonary nodule. What does that mean?
I think you did surgery.
Yeah,
You found that the node, it was
She had nodes.
Yeah,
Yeah,
she had
surgery
you missed it.
and a CAT scan that's
Yeah, they didn't get all the periodic nodes out.
Yeah.
Yeah, Matt
In,
left
that,
a
in
note.
this case, well, if she's not asking for a trial. She
Right.
would be a perfect patient to be given chemotherapy plus immune checkpoint inhibitor. We don't have anything better yet,
Chemo,
PMMR, right?
even if it is
chemo plus IO.
yes,
Ah.
chemo plus IO. Well, now it may be my bias. I can't say that, but in that case, maybe. Well, I don't have the other status of this trial. It's yeah, at least PMMR IO I would be adding. I'm not sure how we are going to deal with the approval from EMA on the addition of PARP inhibitor.
Yeah. Well, I was going to ask you, would you offer her a PARP as well? What would you recommend? She says, I read this trial that was just presented.
So let me put
ITCS,
it this way.
ESMO.
Let me put it this way. And now again, I'm looking at the non-analytical hazard ratios of
And make a
subgroup
decision
of
for
a
treatment.
subgroup to take a decision. I just said that you should not do that, but I'm going to
I'm
do
glad
that.
you're doing it.
So if you look at the molecular classification of Ruby Part 1, where only immune checkpoint inhibitor was added, tistalimab, in the p53 mute population, you had, and this is p53 mute, grade 3 endometrial.
We don't know.
We don't know.
Probably, most probably. If it is, the hazard ratio was 0.55. If you add in the duo E, PARP inhibitor on top of that, hazard ratio is 0.47. Completely criminal. I'm trying to do the trans-trial comparison. I'm doing the subgroup of a subgroup, all that. And the toxicity will be added. I'm not sure that I'm there to add another drug, but if there is an approval, this is a possibility, and probably some of my colleagues will do that.
Yeah, I mean, we do have a trial it's analytical for a PMMR subgroup like this, right?
Yeah.
018. I mean, you could use that as a
Exactly.
as reference point for. I mean, I do think we can do better here. I think if this was actually a p53 wild type, then that would raise the question, as I mentioned, for the export. And full disclosure, I'm the USPI for export EC. But that would be another thought if you were uncomfortable using an IO or didn't have access to it. That would be an opportunity, even though she declines the participation on a clinical trial. That could be done as a maintenance.
So what about same case, HER2 3+?
Oh, then I would move to US.
Well,
Or to include the patient in the clinical trial,
Yes.
Yeah, but she doesn't want it.
She doesn't
trial.
want clinical trial.
You have to give her best recommendation.
because she never seen you, Monso.
Best recommendation is clinical trial. No, I mean, but in the U.S. though, she would be eligible for trastuzumab with chemotherapy because she's 3+.
So what if we take her back just a little bit?
That's in the
to
NCCN
3C
guidelines.
She's now 3C1, same criteria, HER2 negative, and would have been eligible for B21, EN11. So
So-
just slight difference in where the lymph nodes were located, but we took all the lymph nodes out. She's non-measurable. Now you tell her no
Is she
chemo, no
PM
IO.
Is she PMMR?
Yeah.
She's
Yeah.
PMMR.
I'm not going to treat her.
Because they actually look like they do worse when you give them checkpoint, right?
I think
So
the
a
bio-
difference
I
of
would-
one
yeah.
lymph node higher,
Strong.
you recommending IO therapy. But if it was just
No,
in the
that's not the
pelvis,
case. That's not the, I'm talking about there is no residual disease. That's the biology, and it's a PMMR. Probably it will be, well, if there is a regulatory approval, then again, you will get it. But that's the group. If she was DMMR, I have no question. I will add immune checkpoint inhibitor here, whatever is 3A or 2 or even 1.
So if you don't give her an IO, what are you giving her?
That's the
I
problem.
mean, if you wouldn't give her what?
Great.
You're not going to give her an IO,
Uh-huh.
checkpoint inhibitor.
Uh-huh.
How are you going to treat her? She's now no residual
3C1,
disease.
no
No,
residual
we're going to make her no residual disease.
PMMR.
We just changed it. That's it.
yep, PMMR.
And what would you do?
I mean, if you could do chemo alone, this is a very small subgroup. I cannot answer that. But if you look at the whole PMMR in the Ruby, you have
Quickly,
added
what would you do?
7
She wants
months
to know.
of overall. Now I'm contradicting myself. 7 months of median OS in this population. But that's a huge population. So I don't know how I'm going to distract. I would suggest what the best is, talk with the patient, and probably still there is a place until Dr.
Yeah,
Coleman
she went for
does
a
his
She went for a
trial.
consult. She went for a second.
What about
opinion.
Isabel? What, 3C1,
What do
non-measurable,
you think of Isabel?
lymph nodes removed from the pelvis, that's all she has. What's your recommendation for that patient?
I will be in favor. She has only 54 years old. She's very young.
Yes.
So we have to try to cure the patient.
Right.
So I will try to move in the way to give her access to immune therapy. However, we need to understand why she declined to participate. It's because she's fine.
She had breast cancer 2 years ago.
Yeah,
She's not
okay,
eligible.
Okay. Breast cancer, which one? Triple negative? Because if it is triple negative, I will push in favor to also add immune therapy if she did not receive before.
Yeah.
No,
No, she did not have any immune
we have to
therapy.
say that, unfortunately, in Europe, we can do what we want. If the patient can benefit of what we published with the previous trial, we will move to give immune therapy in this patient. If it is not feasible, I don't do it.
In the PMMR in Europe
Yeah.
right now, we have only a possibility to give together with a PARP inhibitor. That's the approval. We don't have of approval of chemotherapy plus immune checkpoint
But I have
inhibitor in the
accelerated
PMMR. It will come, but right now, if this is the case, that is the treatment for this patient.
Any role for radiation in this patient?
Oh, no.
I know, I was going to ask the same, yeah.
Well, I'm glad I didn't hear it from either of them, so we didn't have to refute it. But with the OS on GOG 258,
Yeah.
just not showing a trend towards
Yeah.
benefit,
Right.
it's, a 54-year-old, that's a long time to suffer from some of the
She's
radiation.
endometrioid. She's not papillary serous.
Right,
She's not serous. She's not.
I mean, you can consider it,
chemo.
but I
Yeah.
think it's going to provide some local control.
I mean, the tumor board, the radiation oncologist is
For sure,
definitely
say
going to
something.
recommend radiation to that.
How many,
to that
you
patient.
want to ask the audience how many people just give chemo?
Yeah, how many would just give chemo alone?
Yeah.
There's no wrong answer here, as you can see. Our panel didn't even want to answer it. How many would give chemo plus IO?
If I had
How many people would do some type of combination of chemo and radiation? Fair number, yeah.
Yeah. Well, it's about a third, a third, a third.
Yeah.
Yeah.
Yeah, interesting.
Anything else?
Even our
Bring it all
medical
together.
oncologist.
All right. I want to thank everyone. I want to thank our presenters. It was an outstanding job. Thank you so much. You did a great job. The session will be available on the IGS on-demand platform after the meeting. And again, we want to thank GSK for support of this symposium. I hope you have a great afternoon.